Hello everyone, today we're going to look at tissue fixation. Fixation comes from the word fixative, and fixative is a substance which preserves the shape, structure, relationship, and chemical constituents of tissues and cells after death. Therefore, fixation is the process of maintaining tissue or cell morphology in a lifelike state as much as possible. And fixation usually helps prevent postmortem changes. And among the postmortem changes that can occur on a tissue or cell, we have autolysis and putrefaction. So usually when there is blood interruption, there are changes that will take place because the tissue or the cells are no longer receiving the nutrients direct from the body. So these postmortem changes take place on a cell or tissue after death of an organism. So they can occur in any tissue, for example, skin, breast, liver, so on and so forth. And the most prominent postmortem changes in this case, we have autolysis and putrefaction. Autolysis, to be specific, refers to destruction of cell or tissue. But this destruction of a cell or tissue is caused by enzymes, and these enzymes are produced by the lysosomes. So these enzymes digest away the different cellular components following death. The other prominent post-mortem change that can occur after death is putrefaction. And the putrefaction refers to destruction of cell or tissue by normal flora in normal and pathological conditions. So because of putrefaction, we are going to have production of a gas which leads to a filthy smell. So those are the most common changes that can occur after death. And postmortem changes are prominent in highly specialized tissues because of their structure and functions. For example, we have the liver, the intestinal tract, kidney, and the brain. Therefore, by fixing the tissue, we are trying to avoid these postmortem changes, because when they occur and we process the tissue up to the level of staining, we are most likely going to report false positive results, because these changes under the microscope, because of these changes, certain things will appear to be abnormal within the tissue, when actually they were caused by, or they came as a result of not fixing this tissue immediately after blood interruption. Therefore, it is very important, upon blood interruption, tissues or cells should be fixed immediately. In our subsequent discussion, we shall look at the purpose or the various functions of fixation, or the various functions of fixing tissues immediately after blood interruption. Thank you very much for listening.